How?
Genetic risk converges on regulatory networks mediating early type 2 diabetes. Hey everyone, and welcome back to the channel. Today, we're diving deep into the mysteries of type 2 diabetes, a chronic condition affecting millions worldwide. But this time, we're not focusing on the sugar spikes or the insulin woes. We're venturing into the microscopic world, where genes and their intricate dance with cellular machinery are key to understanding the disease's early stages. Buckle up, because we're about to unravel a groundbreaking study. In this study, researchers investigated the molecular and functional changes in the pancreatic islets of donors with early-stage type 2 diabetes, T2D. They used a multimodal approach to integrate data from islet function, tissue architecture, and cell type-specific gene expression to understand the disease process. The study revealed that early T2D is characterized by intrinsic defects in beta cells, reduced insulin secretion, and alterations in the islet microenvironment. Furthermore, the research identified the regulatory gene RFX6 as a key player in T2D pathogenesis, with multiple layers of genetic risk converging on its gene regulatory network. Diabetes is a complex and prevalent disease, characterized by dysfunction of insulin-producing pancreatic islet beta cells. Despite extensive research, understanding the molecular mechanisms behind the early stages of type 2 diabetes, T2D, remains challenging. In this study, researchers aim to unravel the molecular and functional changes in the pancreatic islets of donors with early-stage T2D. By employing a multimodal approach, they sought to integrate data from islet function, tissue architecture, and cell type-specific gene expression to gain a comprehensive understanding of the disease process. Type 2 diabetes, T2D, is a complex metabolic disorder characterized by dysfunction of insulin-producing pancreatic islet beta cells. Although T2D is known to have a strong genetic component, the biological mechanisms underlying the disease, especially in its early stages, remain poorly understood. In this study, researchers aimed to gain a comprehensive understanding of the molecular alterations associated with early T2D by analyzing islet function, gene expression, and tissue architecture. The researchers collected pancreatic tissue and isolated islets from donors with early-stage T2D and non-diabetic (ND) donors. They conducted functional profiling of islets using various treatments and measured insulin secretion. In addition, they performed high-resolution molecular profiling of the pancreata from the T2D cohort, including bulk RNA-seq and histologic analysis. The study also involved the perturbation of the regulatory gene RFX6 in beta cells of primary human pseudoislets, allowing for functional, transcriptomic, and epigenomic analyses. Furthermore, using Mendelian randomization analysis, the researchers leveraged population-scale genetic data to examine the relationship between reduced RFX6 expression and T2D. The study revealed several key findings. First, the functional analysis of islets showed a reduction in stimulated insulin secretion in early-stage T2D, both ex vivo and in vivo, even in a normoglycemic, non-insulin-resistant environment. This indicated intrinsic beta cell dysregulation and/or alterations in the islet microenvironment as key features of early T2D. Second, the researchers identified the regulatory gene RFX6 as a candidate disease-associated gene, with reduced RFX6 expression associated with T2D. Molecular perturbation of RFX6 led to reduced insulin secretion, defined by transcriptional dysregulation of vesicle trafficking, exocytosis, and ion transport pathways mediated by genome-wide chromatin architectural changes overlapping with T2D genetic risk variants. Furthermore, Mendelian randomization analysis revealed a causal relationship between reduced RFX6 expression and T2D. The functional analysis of isolated islets revealed a substantial reduction in stimulated insulin secretion in early T2D, both ex vivo and in vivo. Transcriptomic analysis showed dysregulated pathways in beta and alpha cells, including those involved in insulin secretion, lipid metabolism, and cilia organization. Additionally, the study found subtle changes in the islet microenvironment, such as reduced capillary size and alterations in immune cell populations. The findings of this study shed light on the complex molecular alterations associated with early T2D, emphasizing the intrinsic defects in beta cells as a predominant feature of the disease. The integration of multiple modalities has provided a comprehensive understanding of the pathophysiology of early T2D, highlighting the need for further research to elucidate the underlying mechanisms and identify potential therapeutic targets. This study isn't just an intellectual exercise, it holds practical promise. By understanding the early-stage conductors and soloists of type 2 diabetes, we can develop better diagnostic tools and potential therapeutic targets. Identifying individuals with specific genetic variations in RFX6 or other key regulatory network genes could enable earlier diagnosis and intervention. Moreover, targeting these network hubs with novel drugs or gene therapy could offer innovative treatment strategies to prevent or reverse early-stage dysfunction. While the battle against type 2 diabetes continues, this study offers a hopeful coda. By illuminating the intricate genetic dance behind its early stages, researchers have opened doors to personalized medicine and potential cures. This study marks a significant turning point in our understanding of early-stage type 2 diabetes. By shifting focus from individual genes to the complex networks they orchestrate, it paves the way for a new era of personalized prevention and intervention. So, the next time you hear the whisper of diabetes, remember the silent heroes of this study, the researchers who shed light on the hidden networks, giving us hope for a future where we can rewrite the story of this chronic disease. Thank you for joining us today. If you found this information interesting, be sure to like and subscribe to our channel for more exciting updates in the world of science and research.
PharmaPal.